In this video, Team 8, Muhammad, Jaden, Stephanie, and Summer, will all go through cellular biology as animal cells, plant cells, bacterial cells, the endomembrane system, cytoskeletons, mitochondria, and much, much more. The animal cell, as seen on the left, is a eukaryotic organism where it has various organelles for highly specific functions. We will be going through the animal cells, very well-known organelles in alphabetical order. First, the centrosome is the organelle near the nucleus which acts as the main microtubule nucleating organelle. It is vital in cell division with genome stability and mitotic spindle orientation. It also deals with cell symmetry and polarity. Chromatin are packaged DNA and proteins, which are histones, within a chromosome for stability. When unraveled, it is usually for DNA replication or gene expression. The cytoplasm is the jelly-like substance within a cell that composes the intracellular space. It helps with cell structure and contains free-floating ribosomes or nutrients within it. It is also the main site for glycolysis, and as well as fermentation. The Golgi apparatus, a part of the endomembrane system, it modifies and sorts proteins as well as lipids. It is also the site of synthesis for most carbohydrates. It has cisternae for transportation and enzymes for modification. Golgi vesicles, aka cisternae, are used to transport molecules in flattened membrane sacs. Intermediate filaments are polymers of proteins that vary depending on cell type. They are a part of the cytoskeleton and are used for structural support. Lysosomes are organelles in the cytoplasm containing enzymes used to break down macromolecules. It has protein pumps to keep the pH at 5 because not only does it help break down such molecules, it is the optimal pH for these enzymes. In this slide on to the left, we can see an actual animal cell. Here, there are not as many labeled organelles, but it is still very useful in how it can be realistically seen under a microscope. Continuing the organelles, microfilaments are helical polymers of actin monomers. They are dynamic and can disassemble or assemble rapidly. They are used for structural support. Microtubules are hollow polymers of tubulin dimers. They are dynamic and deal with dynamic instability. They are also used for structural support. The mitochondria is a double membrane organelle with an aqueous compartment in between. It is the site of the citric acid cycle, electron transport chain, and ATP synthesis, providing the cell with energy. The nucleolus is used to assemble ribosomes. The nucleus contains the genetic material of the cell and is the site of RNA synthesis. It has its own little membrane and is a part of the endomembrane system. The peroxisomes are a single membrane organelle that contains enzymes that break down fatty acids and amino acids. These reactions produce hydrogen peroxide, hence the name peroxisomes. The plasma membrane is a phospholipid semi-permeable bilayer with peripheral and integral proteins embedded in it. It is used for cell structure, to control transportation of molecules, and protection of the cell. Ribosomes, which can be located in the cytoplasm, in the membranes of multiple organelles as well, they control protein synthesis and translation. In eukaryotic cells, such as the animal cell, they are ADS. Here on the left, we can see a largely stylized version of the animal cell. It is also a little bit more minimalistic in terms of how much is really in the cell. Continuing the organelles, the rough endoplasmic reticulum is connected to the nucleus through membranes, forming the endomembrane system. It has ribosomes in the membrane, which helps produce proteins. It also transports such proteins. Secretory vesicles are vesicles that transports molecules into the cell, like hormones or neurotransmitters. The smooth endoplasmic reticulum is connected to the nucleus through membranes, and it produces and transports lipids. Vacuoles absorb and stores water. It also contributes to turgor pressure. However, in animal cells, they are much smaller. Structures only found in animal cells are centrosomes and lysosomes. Now on to the plant cell. To our right, we will see our main diagram, which provides all of our organelles that we will mention today. First of all, the cell wall. As you can see to the right, it is the big, blocky green outer layer. It is primarily composed of cellulose, which is a large carbohydrate composed of many stacked glucose. It is used to filter materials. It is for structure and protection. Then chloroplasts. It is a double membrane structure with stroma as the middle, and then thylakoids, which are internal membranes with pigments like chlorophyll, that are inside of it. They are used to capture energy from sunlight and undergo photosynthesis. Plant cells also have cytoplasm, which is a common theme in terms of cells. Cytoplasm is a necessity for living. Then there are filamentous cytoskeleton. Filamentous proteins which form up the cytoskeleton, and they provide structural support. Golgi vesicles, much like animal cells, are used to transport molecules in flattened membrane sacs. Then also like animal cells, plant cells also have mitochondria. This is because mitochondria produce energy while chloroplasts make energy for storage. As we can see to the slide on the right, here are a variety of plant cells with quite a lot of chloroplasts to absorb as much sunlight energy as they possibly can. Plant cells have a nuclear envelope, which is a 2-lipid bilayer membranes that surround and protect the nucleus. They also have a nucleus and a nucleolus as well, much like the animal cells. They also have plasma membranes, other than just their cell wall, for protection and also to control what comes in and out. They have plasmodesmata, which are small channels that connect plant cells through their cytoplasm. They also have peroxisomes as well. And they also have ribosomes, which are essential for life. Here to the right, we will see a much more stylistic, minimalistic plant cell diagram. Although, unlike the minimalistic animal cell diagram, it has a lot more inclusivity in terms of organelles. Much like the animal cell, plant cells also have a rough and smooth endoplasmic reticulum. This is because multicellular organisms and eukaryotic organisms in general require these in order to fully carry out their duties as a cell and to survive. They have small membranous vesicles, a small sac used to transport molecules around the cell. They also have vacuoles, but they are much larger in plants because water has a huge role in photosynthesis. The structures only found in plant cells would be chloroplasts, cell walls, and the very large vacuoles. And to repeat why there's a mitochondria and a chloroplasts are used to store energy, and mitochondria are used to make energy that are used immediately. Plus, it is very efficient to have multiple ways to make large amounts of energy. And on to the bacterial cell. The bacterial cell is a prokaryotic organism, meaning no compartmentalization, no specialization, and no large organelles. So basically, it is very simplistic, the basics needed for life. As we can see on the left here, it resembles much like a minimalistic version of a diagram for a larger cell, but missing vital organs such as a nucleus. Okay. They have bacterial flagellum. They are made of protein flagellin, and they are used for transportation. They have capsules, a polysaccharide layer that surrounds the membrane, and it is used for support and protection. They also have cell walls, much like plant cells. They are primarily composed of cellulose, much like plant cells, and they have similar functions as well. They also have cytoplasms. This is because cytoplasm is a necessity in order to be considered living, or even live at all. They have DNA in a nucleoid. It is of irregular shape, and it is considered a region of the cell that contains genetic material. Mesosomes are areas in the membrane that fold inward. They have enzymes to break down molecules, and they are a part of cellular respirations. They also have plasma membranes, much like the other cells, a necessity to live. And they also have ribosomes. However, in prokaryotes, they produce 70S.
The mitochondrion is a complex organelle found in eukaryotic cells that provides the largest source of energy for a cell in the form of adenosine triphosphate, or commonly known as ATP. The mitochondrion is also involved in cellular respiration, which is a set of metabolic processes used to convert molecules such as glucose into a usable form of energy for the cell. Cellular respiration includes the metabolic processes of glycolysis, the citric acid cycle, the electron transport chain, and the Calvin cycle. The mitochondrion also has its own DNA and ribosomes. This is because the mitochondrion was once an external organism similar to bacteria. Because mitochondria was separate from human cells, it made its own DNA and translated this DNA with its own ribosomes. Mitochondrial DNA is different from nuclear DNA in that the location of mitochondrial DNA is in the mitochondria, and the location of nuclear DNA is in the nucleus. The mitochondrion contains two membranes, an outer membrane and an inner membrane. The outer membrane is selectively permeable and contains pores that allow small substances such as proteins to come in and out of the mitochondrion. The outer membrane also fully encloses the inner membrane along with the entire mitochondrion structure. The inner membrane is very restrictive, unlike its counterpart, the outer membrane. The inner membrane encases the matrix. As said earlier, the mitochondrion has two membranes. The double membrane nature of the mitochondrion has led scientists to believe that the mitochondrion being a part of the animal cell is a result of endosymbiosis. Endosymbiosis is the symbiosis of two organisms, where one organism lives inside of the other organism and the two function as one. This belief regarding the mitochondrion, as well as the chloroplast, is known as the endosymbiotic theory. The mitochondrion contains many structures, from ATP synthase to the mitochondrial matrix. ATP synthase synthesizes ATP from ADP and inorganic phosphate. This is done using a proton gradient, where the ADP is spun and allows the phosphate group to bind to it, through a process called phosphorylation. The intermembrane space is a space in between the outer and inner membranes. The crista are the folds in the inner membrane that give the mitochondrion a wrinkled appearance. These folds allow for more surface area, which in turn allows for more chemical reactions to take place. Mitochondrial ribosomes are responsible for the synthesis of proteins as well as the translation of messenger RNAs encoded in the mitochondrial DNA. Keep in mind that the mitochondrion contains its own DNA and ribosomes. The mitochondrion also contains granules, mitochondrial matrix, and deoxyribonucleic acid. Granules, also known as matrix granules, are small particles that somehow allow the inner and outer membranes to connect while maintaining homeostasis in the mitochondrion. The function of these granules is a highly researched topic. The matrix is enclosed by the inner membrane, separating itself from the intermembrane space. The mitochondrial matrix is a gel-like substance that provides the site for cellular respiration to take place, a very important biological process composed of a series of metabolic processes. Deoxyribonucleic acid, in the form of mitochondrial DNA, is located in the cytoplasm or matrix of the mitochondrion. Mitochondrial DNA codes for the production of proteins synthesized by mitochondrial ribosomes. The mitochondrion is a widely discussed topic in the science community. Recently, Fenna Henderson and others researched mitochondrial DNA maintenance. They found that EXD2, an exonucleus that has been known to be involved in nuclear double-strand break repair, has mitochondrial localization, specifically in the outer membrane. This is interesting in that it seems nuclear DNA has a connection to mitochondrial DNA, and the role of mitochondria in break repair may not be direct, but a consequence of altered mitochondrial dynamics and/or function. Did you know that mitochondrial DNA can only be inherited from the mother? Mitochondrial DNA allows for the tracing of an individual's family lineage. More and more research is being done on the mitochondrion each and every day through the constant innovation of scientists.
Hi, my name is Stephanie Salazar. Today I'm going to be covering the nucleus and endomembrane system. First, we're going to take a look at the nucleus. Some of the items are going to be covered are spliceosomes, nuclear envelope, nuclear lamines, nuclear pores, nucleolus, euchromatin, heterochromatin, and then we're going to answer some open-ended questions at the end. First, we're going to talk about the spliceosome. It is a large ribonucleoprotein. Spliceosomes remove introns from transcribed mRNA, a type of primary transcript. After introns are removed, exons are ligated together. Introns are removed because they are not needed or contain incorrect material that could compromise the cell's integrity and growth. Spliceosomes are composed of 5 small nuclear RNAs, snRNAs, as well as proteins. The snRNAs bind to the proteins to produce small nuclear ribonucleoproteins. Now we're going to move on to the nuclear envelope. It surrounds the nucleus with a double membrane of lipids with multiple pores. The nuclear envelope keeps the contents of the nucleus, called the nucleoplasm, separate from the cytoplasm of the cell. The separation makes it possible for chemicals in the nucleus to 1. Prepare for and take part in replication of genetic material prior to nuclear division, and 2. Manufacture different types of RNA for export to cytoplasm. The nuclear envelope is more or less a protective barrier for the nucleus. Next up is the nuclear lamines. The nuclear lamina is a dense, fibrillar network inside the nucleus of most cells. It is composed of intermediate filaments and membrane-associated proteins. It provides support for the nucleus and regulates important cellular events such as cell division. The nuclear lamines are located on the inner face of the bilayer nuclear envelope. Our next topic is nuclear pores. These pores regulate the passage of molecules between the nucleus and cytoplasm, permitting some to pass through the membrane, but not others. Nuclear pores are fully permeable to small molecules up to the size of the smallest proteins, but form a barrier against most large molecules out of the nucleus. There are aqueous channels generated from a complex network of evolutionary conserved proteins known as nucleoporins. Next up is the nucleolus. Nucleoli are made of proteins, DNA, and RNA and form around specific chromosomal regions called nucleolar organizing regions. It is best known as the site of ribosome biogenesis, where ribosomes are formed. The nucleolus helps to synthesize ribosomes by transcribing and assembling ribosomal RNA subunits. These subunits join together to form a ribosome during protein synthesis. Accordingly, the size of nucleoli depends upon the ribosomal requirements of the type of cell in which they are found. In cells that produce large amounts of protein and thus call for significant numbers of ribosomes, the size of the nucleolus is considerable. sometimes occupying as much as 25% of the total volume of the nucleus. Now let's talk about euchromatin. Euchromatin is the genetically active region of the chromosome. It contains the structural genes that are replicated during G1 and S phase of interphase by allowing polymerases to access the genes. It also allows RNA gene regulatory proteins and RNA polymerase complexes to access these genes to be transcribed into mRNA products. Euchromatin participates in the active transcription of DNA to mRNA products. The unfolded structure allows gene regulatory proteins and RNA polymerase complexes to bind to the DNA sequence, which can then initiate the transcription process. Our last topic is going to be heterochromatin. Heterochromatin is a form of chromatin that is densely packed and is found in the nucleus of eukaryotic cells. Heterochromatin is in such a condensed structure that it does not enable DNA and RNA polymerases to access the DNA, therefore preventing DNA replication and transcription. There are two types of heterochromatin, constitutive and facultative. Constitutive heterochromatin is a stable form of heterochromatin, just meaning that it does not loosen up to form euchromatin and contains repeated sequences of DNA called satellite DNA. It can be found in centromeres and telomeres and usually involved in structural functions. Facultative heterochromatin, on the other hand, is reversible. Its structure can change depending on the cell cycle and is characterized by another kind of repeated DNA sequence known as line sequences. Now we're going to answer a few open-ended questions. What is the endomembrane system? What does it do? And why is it important? It is a group of membranes and organelles in eukaryotic cells that work together to modify, package, and transport lipids and proteins. It is important because without the transport of lipids and proteins into the body, certain functions would not be feasible. For example, in muscle cells, a special type of smooth ER, which is included in the endomembrane system, called the sarcoplasmic reticulum, is responsible for the storage of calcium ions that are needed to trigger the coordinated contractions of the muscle cells. Question number 2. Describe how a protein destined to be secreted via exocytosis is made. Proteins for exocytosis can be located on the surface of the rough ER. The proteins are synthesized and collected in the cisternal space/lumen sacs on the rough ER. Once synthesized, they are pinched off into secretory vesicles. These vesicles have similar membranes to that of the plasma membrane, allowing them to fuse into the plasma membrane and release its contents. Question number 4. Describe how lysosomes are made. Lysosomes are manufactured and budded into the cytoplasm by the Golgi apparatus with enzymes inside. The enzymes that are within the lysosomes are made in the rough endoplasmic reticulum, which are then delivered to the Golgi apparatus via transport vesicles. When lysosomes reach the cytoplasm, fusion forms a secondary lysosome. The next section we're going to be talking about is the endomembrane system. A few of the topics that we're going to go over are the Golgi apparatus, the plasma membrane, the lysosome, secretory vesicles, smooth endoplasmic reticulum, rough endoplasmic reticulum, the nucleus, and ribosome. The nuclear pore and nuclear envelope were covered in the previous nucleus section, so you can just refer to those slides for that information. The first topic we're going to be talking about is the Golgi apparatus. The Golgi apparatus packages proteins into membrane-bound vesicles inside the cell before the vesicles are sent to their destination. The Golgi apparatus is made up of a series of compartments consisting of two main networks, the cis-Golgi network and the trans-Golgi network. The CGN is a collection of fused, flattened membrane-enclosed discs known as cisternae, originating from vesicular clusters that bud off the endoplasmic reticulum. The TGN is the final cisternal structure from which proteins are packaged into vesicles destined to lysosomes, secretory vesicles, or the cell surface. Up next is the plasma membrane. The plasma membrane regulates what enters and exits the cell, as well as helps maintain the cell's shape and integrity. The cell membrane is primarily composed of proteins and lipids. While lipids help to give membranes their flexibility, and proteins monitor and maintain the cell's chemical climate and assist in the transfer of molecules across the membrane. The lipid bilayer is semi-permeable, allowing certain molecules to diffuse across its membrane. Plasma membrane is an amphipathic, which contains both hydrophilic heads and hydrophobic tails. It is a fluid mosaic of lipids, proteins, and carbohydrates. It is lipid bilayer, which contains two layers of phospholipids. Phosphate head is polar, water-loving, and the fatty acid tail is nonpolar, water-fearing, and the proteins embedded in membrane. Now we're going to discuss the lysosome. Lysosomes are spherical vesicles that break down biological polymers, such as proteins, carbohydrates, and lipids. It is composed of membrane protein and proteins of its lumen. The lysosomes act as the waste disposal system of the cell by digesting unwanted materials in the cytoplasm, both from outside the cell and obsolete components inside the cell. Material from outside the cell is taken up through endocytosis, while material from inside of the cell is digested through autophagy. Lysosomes also are involved in plasma membrane repair, cell signaling, and energy metabolism. The lipids that make up the bilayer of the lysosome are phospholipids, which are molecules that have hydrophilic phosphate group heads, a glycerol molecule, and hydrophobic fatty tails. There are many different types of enzymes in lysosomes, including proteases, amylases, nucleases, lipases, and acid phosphatases, among many others. To follow up is a secretory vesicle. Secretory vesicles are small membrane-bound sacs that contain materials such as hormones or neurotransmitters, which are packaged together by the Golgi apparatus and eventually released via exocytosis. These vesicles have similar membranes to that of the plasma membrane, allowing them to fuse into the plasma membrane and release its contents. The vesicle can also fuse to other organelles within the cell. Cells have many reasons to excrete materials. One reason is to dispose of waste. Another reason is tied to the function of the cell. Within a larger organism, some cells are specialized to produce certain chemicals. These chemicals are stored in secretory vesicles and released when needed. Our next topic is the smooth endoplasmic reticulum. The smooth ER helps synthesize phospholipids and cholesterol. The network of smooth endoplasmic reticulum allows for an increased surface area to be devoted to the action or storage of key enzymes and the products of these enzymes. General functions include the folding of protein molecules in sacs called cisternae and the transport of synthesized proteins and vesicles to the Golgi apparatus. The smooth ER also regulates and releases calcium ions and processes toxins. Smooth ER also makes or distributes products according to its location. In muscle cells, it releases calcium ions because they are required for muscle contraction. In cells of the endocrine system, which is responsible for regulating hormones, smooth ER makes steroid hormones. The smooth ER is tubular in shape. Next up is the rough endoplasmic reticulum. The surface of the rough ER is studded with proteins, giving it the rough appearance. The RER is generally a series of connected flattened sacs. It plays a central role in the synthesis and export of proteins and glycoproteins. The ribosomes attached to the rough ER synthesize proteins by the process of translation. Question 2. What is the importance of nuclear pores? Nuclear pores are important because they regulate what goes in and out of the nucleus between the cytoplasm. Small particles can go in and out as they please. However, most large particles are kept from the nucleus. Without the pores regulating the nucleus's contents, the entire cell could be compromised, and the nucleus could be introduced to many dangerous particles and substances. The nucleus is the brain of the cell. This lack of regulation could lead to the cell's eventual failure and death. Our last topic to be covered are the ribosomes. Ribosomes are composed of ribonucleic acid and proteins in nearly equal amounts. The ribonucleic acid is derived from the nucleolus, where ribosomes are synthesized in a cell. Some ribosomes are found scattered in the cytoplasm, referred to as free ribosomes, while others are attached to the endoplasmic reticulum. They play the role of assembling amino acids to form specific proteins, which in turn are essential for carrying out the cell's activity.
Which are made of linear polymers of G-actin proteins. They are double helix shaped and act as a track for the movement of myosin molecules. Intermediate filaments, more stable than microfilaments, and they organize the structure of the cell. Microtubules, which are hollow cylinders that bind GTP for polymerization, organized by the centrosome, and form centrioles, cilia, and flagella. Septin, GTP binding proteins that are conserved, may form filaments and rings, and may serve as an attachment site for other proteins, preventing certain diffusions. And spectrin, that lines the intracellular side of the plasma membrane, has both pentagonal and hexagonal forms. Prokaryotic cytoskeletons consist of FTSZ, which form filaments when GTP is present. When cell division occurs, FTSZ is the first protein in the division site, and it is needed to gather other cells to create a new cell wall. mREB, and PARM, which maintains cell shape, and also chrysantine, which maintains cell shape.
And here is our APA citations. Hopefully, you learned something.